Okay, we'll get going then. Welcome again, and thank you all for dialing in tonight to the latest 2022 edition of State of Play. For those of you on Zoom, you'll see a notification about the meeting being recorded. Just click OK or agree, and that should go away, that little notice. So first of all, an acknowledgement of country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islander peoples on this call today. So today's theme is around changes in the brain during MND. Our first presenter is Dr. Rosie Clark from the University of Tasmania. Rosie's a former Bill Gold Fellow funded by the MNDRA, which is our most prestigious fellowship. And Rosie's work delves into the sort of cellular and molecular changes that happen during the brain during MND. Second up, we have Dr. Thanuja Dharmadasa. Sorry for any butchering of the pronunciation there, Thanuja. And Thanuja's a neurologist and researcher who's recently returned from Oxford to work at the Florey Institute in Melbourne, and she holds an NHMRC CJ Martin Fellowship. And Thanuja is going to tell us about her work measuring changes in the brain during the disease process. As previously with our sessions, each presenter will talk for about 20 minutes, and we'll have a combined Q&A at the end of the session. If you have any questions, please fire them in either through the chat or Q&A function if you're on Zoom, or if you're watching through the Facebook live stream, you can send us your questions through the comments section there. So that's enough from me. So please take it away, Rosie.
Okay, I'll just begin with sharing my screen would be a great start. Okay. Okay. So first of all, I'd like to thank Ethan and Laura for inviting me to give the talk today in the State of Play series. And today, as Gethin mentioned, I'm going to be talking to you a bit about the changes in the brain, focusing on some of the molecular and cellular changes. And in particular, I'm going to be telling you a bit of a story about some research we've been doing and how we can get important clues from some of the clinical heterogeneity we see in the disease, and how, I guess, that clinical heterogeneity is associated with changes that we see in terms of the cells and the pathology that changes in the brain with people with MND, or MND ALS, as I'll probably be saying throughout the talk. So I think this fellow here really surmises, I guess, where my research is focused, and it's really looking at the balance of neurotransmitters in the brain of people with MND. And increasingly we learn more and more that there's changes in this balance between the two key main neurotransmitters in people with MND, particularly in the motor cortex. And when this balance becomes disrupted, obviously there's detrimental consequences. So I'm gonna, there we go, click on. So I'm talking to you about determining the role of a particular peptide in ALS or ALS-MND. So what are the mechanisms underlying motor neuron disease, and where does it all begin? These are two main questions that I guess the field has been grappling with for quite a while. And there is a number of different mechanisms that we know at a cellular level are quite important for the motor neuron, its health, function and survival. And there's a number of these factors here that become affected in the disease with different things like endoplastic reticulum stress, altered axon transport, mitochondria, which affect how these motor neurons function. So these axon-like structures are huge in a motor neuron, and they act as a highway basically to send these connections all the way down to our muscle. So they're quite vulnerable in the disease. But there's a number of different ways that these motor neurons are vulnerable. And in particular, I'm interested in this process called excitotoxicity. So motor neurons have these receptors on them that basically respond to glutamate. And when there's too much glutamate around, the motor neurons become sick and they can degenerate. And basically the connection to the lower motor neuron, which is residing in your spinal cord, is lost. And then the connection forward as well to the muscle is lost. So when you've got these upper motor neurons in the brain, in the motor cortex, that change their function, and you have the spinal cord lower motor neurons that then feed that signal forward, becoming degenerative and losing their connections to the muscle, then you begin to see muscle loss and atrophy. So a lot of my research has been focusing in the brain, looking at what happens to these upper motor neurons and what changes in the brain, in particular in regards to this concept, which is called excitotoxicity, but it just equates to too much glutamate in the brain. So there's a lot of evidence at the moment that there's a lot going on in the brain with people with MND. So we know there's changes in neuroinflammation, we know there's changes in mitochondria, all of those processes that I mentioned earlier. And there's evidence that this is occurring for a long time before symptoms become apparent in people. And we kind of want to get an idea about what are the main factors that influence, I guess, this final emergence when a person suddenly starts to present with these symptoms of muscle wasting and progression. And what we're learning from clinical evidence from brilliant groups up in Sydney is that there's a change in the activity within the brain of people with MND. So what you can see is there's this rapid hyperexcitability that occurs inside the brain of people with MND. And this occurs in both forms of the disease. So whether you've got a familial genetic inheritance of the disease or whether it's sporadic and you've got no family members who've been diagnosed, there's this emergence of hyperexcitability in the brain. And this occurs, importantly, months prior to the symptom onset. So imagine you're living your whole life without MND, then the first clinical thing that we can see is actually this change in excitability within the brain. And importantly, this occurs before we see any measurable lower motor neuron dysfunction. So there's no changes that we can detect in the muscles yet. And importantly for my research, we see this drop in inhibition. So excitation is basically sending signals throughout the body, but inhibition is important for providing that balance to those signals to stop too much glutamate from appearing and from stopping those motor neurons from burning out. And so importantly, what they see in the clinic is when you have too much of this excitation, you also have not enough inhibition. And importantly, this has been associated with survival in patients as well. So when inhibition goes down, people don't survive as well. So as I mentioned, excitotoxicity is basically a process that kills neurons through too much glutamate. And as I've mentioned, motor neurons, an excitatory cell, they rely on glutamate to function. So they require it, we need it there, so we can't just get rid of it. But in order to basically stay healthy, they rely on this population of cells called inhibitory interneurons. And these cells are basically the brakes on excitability. So without these guys, we can't control excitability. So a lot of work's focused on these excitatory motor neurons, obviously in disease, because we know they're important for motor function. But less is known about what I call the brakes on this inhibition, these interneuron cell populations, and why they aren't able to keep this glutamate excitability in check. And that's been the focus of a lot of my research. So a lot of people typically think about inhibition and excitation in a normal brain as quite balanced. But what we see in ALS, as I've mentioned, is a decrease in this inhibition and an increase in excitation. And this is associated with the time roughly that symptoms become apparent in people with MND. And this raises the question of whether this decrease in inhibition is actually quite important for instigating that motor symptom loss. So as I've mentioned, you've got these inhibitory interneurons, and a lot of people, even in the neuroscience field, who are less obsessed with interneurons than I am, typically think about them as one cell type that relies on a neurotransmitter called GABA. But in reality, there's a range of these different interneurons in the brain. And In fact, it actually looks more like this. So you can see it's quite a complicated puzzle. And interneurons, in fact, provide a lot of the diversity of function that higher mammalian brains have. And they're really important for controlling that network flow of excitability. And if this is our motor neuron and how it basically has a flow of information coming in and a signal coming out that goes down to our lower motor neuron, you can see in here that there's a range of different interneuron populations that help to connect to different parts of that motor neuron to help it signal and maintain its health. So it's a very complicated but interesting system in terms of motor function. And how I think about interneurons is this guy here. If you don't have interneurons, how are you going to control all those motor networks, coordinate them, to fire at the right time, to tell your muscles to balance? If interneurons aren't there, then this can't happen at all. So they're incredibly important for controlling not only the neuronal excitability within our central nervous system and brain, but also in motor function. So in my early research, in my honours year, in fact, I began looking at this problem and asking what's going on with these inhibitory interneurons. And this is just an image here of the brain in a rodent model of MND. And basically what I'm looking at here are a number of these different groups of interneurons that you saw here. So I'm looking at different subtypes of these interneurons because they all have slightly different functions in the brain. And I'm asking the question, are they changed? Because essentially what we see in the clinic is reduced cortical inhibition. So my hypothesis was that you should see decreased numbers of interneurons within the brain. So I'm looking at these different subtypes to see if they're changed. And what I actually ended up finding was indeed there's a decrease in a certain interneuron population. And in this mouse model, you can see as the disease progresses, there's more of this particular interneuron cell type being lost that just happens to be labeled with a peptide called carotinin. But completely opposite to what I was thinking, I actually found this weird population of cells, and it's always what you're not looking for is what you learn from, called neuropeptide Y labeled cells. And actually what I saw was that there was an increase in the number of these cells in the motor cortex in this ALS model. And that was not at all expected, because what I was expecting to see was this decrease, which I had seen in a certain interneuron population, and maybe this population is important for losing that inhibition. But my main question was, what is this different population doing? Why is it increased in this motor model when motor symptoms are progressing? So looking at the literature, basically I had some reading and I found out these carotinin cells are unique in that they control a lot of those interneurons within the motor cortex, and they can have a wide-ranging impact on overall inhibition. So that made sense in terms of finding. But MPY, or these MPY-labelled interneurons, are very unusual. In fact, they're quite well known in the epilepsy field. And what they actually have found in there is that these population of cells have anti-excitability properties or anti-epileptic, and actually can be neuroprotective in some situations. So in fact, there's increased synthesis and release of this neuropeptide Y compound after people have epilepsy or in models that simulate epileptic seizure events. And in fact, when people apply the peptide that this interneuron produces, they can actually dampen down and reduce this hyperexcitability. And this was very interesting because, yes, it's great to understand how the disease may be progressing, but it's also interesting to look at what's going on in terms of compensatory mechanisms that might be trying to protect the motor system. So I did some further work looking at this NPY labeling to see if it was similar in people's brains with MND. And what you can actually see here is this a control brain of someone who's died from another age-related disorder. And you can see here, this is what the MPY labeling looks like, but this is in ALS. As you can see here in the primary motor cortex, which is where we're looking at that excitability, there's a lot more of this neuropeptide Y present. So this is showing basically that there's an increase in this MPY. And that was very similar to what I saw in that mouse model. because mouse models are important. We can learn a lot from them, but we also have to check to see if this is relevant to what we see in ALS patients. So that's very interesting, as it points to something that's quite important for the disease. And basically from here, that was looking in a few cases. I wanted to extend upon these findings and ask the questions about what this MPY can tell us about the disease processes in MND, and in particular, what's going on in the MND brain. So in that previous ALS brain tissue I looked at, it was very informative, but I didn't have any clinical information available to me about the cohort in terms of the clinical characteristics. So there's important things that we have to look at in regards to MND, and basically how it can progress differently in different people. For example, we know that there's more males who get MND typically than females. There seems to be a sex effect there. We know that people can get MND at different ages of their life, although typically in their later years. And there's variation in where the initial symptoms appear in people, such as bulbar onset MND, where it begins in the tongue with difficulty swallowing and speaking, or limb onset, and it can even vary further, where it starts in the upper limbs, such as in the arms, or in a lower limb, in the legs. And there's also variation in terms of how the disease progression rate occurs once symptoms have appeared. So there's a number of different factors that are different in people, and I wanted to know if this MPY could tell us something about the disease processes in terms of how this clinical heterogeneity appears in people, and importantly, is there something this MPY is doing that might be involved in that excitability? So I asked the question, does NPY associate, or these MPY interneuron that are labelled with any specific clinical characteristics of ALS MND people? And basically I did this cool thing where you can label a protein in the MND brain here, and you can see here that this is the MPY labelled, and this is just a black and white image representing that MPY labelling. So I can create a binary image here and basically look at the extent of that MPY present in the brain, score it, and then look at it relative to known clinical characteristics of people who've passed away from MND. So what I found is that there seems to be, again, an increase in MPY, and this was important to confirm in a larger cohort of patients. As you see here in controls, it's much lower, that MPY expression that I showed you, compared to ALS cases. So we know it's there, but as you can see, there's quite a bit of variability here in terms of the amount of MPY that's present in different people's brains. So I had a look at my tissue again and looked at and made the observation, I guess, of what was going on here. And I saw striking differences in terms of the disease duration that people had experienced from symptom onset until they'd passed away. As you can see in the controls here, we've got some normal labelling of MPY, and then in people with ALS, we have fewer labelling of this MPY, but we have some of those processes apparent. But as you can see here, someone who survived for 60 months with MND, which is unfortunately a relatively long time in terms of MND progression, has an incredible amount of MPY present in their brain. So I'm thinking, okay, that's quite interesting. Perhaps it's associated with the rate of disease progression. So looking at my data, I then went and had a look, doing statistical tests at these range of different factors that I was talking about. And in fact, as we can see here, disease duration popped out as a very important correlate for this MPY signal. And that tells me that basically people who live longer have more of this MPY present in their brain from when they're diagnosed. So that points to a very potentially important role of MPY in the disease. So I went on to do what's called a survival analysis, looking at just how long people had the disease from symptom onset until death, and had a look at how the MPY affected that by basically splitting the groups based on that, the extent of MPY expression. And people had lower MPY expression had a quicker disease progression rate, and people who had more MPY had a slower disease progression rate. So again, it's telling us that this peptide is quite closely associated with how quickly disease is progressing, but it had no difference if you look here in terms of when people basically developed MND. So it seems to be more important for once MND is diagnosed, this peptide is doing something in terms of how the disease progresses. So we learned that MPY appears to be increased and is associated with disease, and it has no other association with any other important clinical characteristics. So it's argument for a role of this particular peptide in disease progression. And importantly, another group up in Sydney has confirmed the presence of elevated levels of this MPY in blood plasma level, because all good science needs to be done as a collective, and we find the same results. It really shows confidence in our finding that this is also related to disease duration in this study. So we're asking the question, maybe MPY is neuroprotective, and there's a lot of evidence to suggest it can modulate a lot of these pathways that we know are involved in MND. And in particular, because of the epilepsy research, I was really focused on this idea about excitability and excitotoxicity, and maybe MPY has an effect on that excitability that we see that is so relevant to disease appearing in people with MND. So my PhD, Courtney, who I call the MEA Queen, who has used a technique called multi-electrode arrays, and basically she applied this neuropeptide Y into cells in a dish to have a look at how it affected the excitability of cells. And what she found is that in diseased cells, there's much higher activity, but when you apply this MPY, it does suppress it. So there's reduced levels of the firing when MPY is applied, as you can see here, which tells us that MPY might be working on the excitability of neurons. And following on from that, we want to know how it affects those actual cells, those inhibitory interneurons, and the excitatory cells, or the motor neurons and the pyramidal neurons in the brain. And what we find is that MPY can actually reduce the excitability of these neurons. So when we actually patch the cells and have a look at their actual activity, we can see that the MPY is reducing that, but it's also increasing the excitability or the function of those inhibitory neurons. That might be how, if it is keeping people alive for longer, potentially how it's having its mode of action. And importantly, a recent study has come out that basically has looked at this reduced inhibition that I mentioned in the ALS brain, and they've shown here that basically as the disease progresses, there's more and more of this cortical inhibition lost over time. And similarly, when we look at our data in MPY, we see the opposite. So as disease progresses, there's more of MPY available, or more MPY in the brain. So this points, again, more evidence that MPY could be more modulating excitability. So in summary, this is just, I guess, a little look into how pathology and how looking at the clinical heterogeneity in the disease can tell us about pathways that are possibly very relevant to disease, particular disease aspects in the MND pathogenesis. And it's very helpful to learn that excitability remains a central disease mechanism in ALS, and potentially doing this work, we can find targets that we can modulate to hopefully protect motor neurons from this enhanced excitability that could be therapeutic, and hopefully slow down disease progression for people with MND. And importantly, though, I think we also learn based on that slope that not all ALS cases are the same. And that's very important. That's where we learn our lessons about what is central to the disease. So I'll just leave it on that slide there. I've probably gone over time, knowing me, talk too much, and just leave it on the thought that balance is important in our brain as much as it is in life. And obviously a cup of tea is quite essential as well. But I think I'll pass it over to Thanuja now I'd just like to thank the team of people who've been involved with this, as well as the people who've donated their brains towards research to help and aid in this research. So thank you very much. And go.
Thanks, Rosie. I'll just share my screen.
I think, yeah, and we've just got Thanuja's about to switch over, and I think she's our clinical equivalent talking about imaging in the MND brain. I'll just stop there.
Hopefully you can see that.
Yep, all good. All good,
Perfect.
Thanuja.
Great. Well, thank you very much. And thank you very much to MNDRA for inviting me to speak tonight. So I'm, as Gethin and Rosie both mentioned, I'm a clinical neurologist and a clinical researcher in MND. I'm recently back in Melbourne. And I'm going to be talking a little bit about the clinical work we've been doing to try and understand this same problem, the changes in the brain in MND. So as Rosie has kind of iterated in her talk, we know that MND involves failure of these two major motor systems, the upper motor neurons that you can see here, which is from the population of nerves from the brain extending down to the spinal cord, and the lower motor neurons that go from the spinal cord out to the muscle. And for us to even kind of to put this into context, for us to even perform a simple activity such as moving a finger, for example, both of these pathways have to be intact and sends messages in the form of electrical impulses from that specific region of the brain down the spinal cord and then finally to contract and move your muscles. So it's the failure of both these pathways in MND that's quite a unique problem. And there's still speculation about where the disease starts, but there's a fair amount of evidence, as you also heard from Rosie, that there's critical involvement of the brain and that probably the disease processes might actually start here. So when we think about the clinical disease, the important role that the brain plays in developing this disease clinically, it may actually stem to some evolutionary observations. And firstly, MND is singularly a human disease. So that really means that it's purely a disease that only humans suffer and no other species really develop it in the way that we do. That also captures why this is a really good way to try and understand, or good way to try and explore the brain and actually understand exactly what's happening in this condition. But when we look at what largely separates us from even our closest primate cousins, it's really in the evolution of how our brain has developed. And we know from historical observations that when we develop these really unique and complex hand use and speech vocalization, we also developed this really increase in our cranial capacity and the development of our brain. And we really increase the complexity of these neurons within our brain. And we predominate with this as a species, as you can see. And so this complex wiring of our motor system, it gave us many skills beyond other species, but perhaps it also gave us a vulnerability to developing certain neurodegenerative diseases. And that's why another reason why the brain is such a critical component to understanding this disease. So how do we actually start to study the human brain? So William Gowers in 1888, so he was a British neurologist, he said the brain is almost entirely inaccessible to direct examination. And that's really been a major difficulty across the centuries and the decades because we're encased in a hard skull and a spinal cord. And it's led to some desperate attempts, I guess, over the years to really look at what's going on under the bonnet. And up until the 20th century, we could really say the brain was almost entirely inaccessible to any direct examination, which is shared in the problem of why this has been really difficult to explore. There's also some very unique clinical features that have made understanding these upper motor neurons or cortical neurons really difficult to understand clinically. And one factor is that the upper motor neuron involvement can be clinically absent in a proportion of patients, or at least stays quite difficult to detect. And that means that it's really hard to quantify that in clinical practice, how much of the brain is involved and how much of involvement of the brain is really driving the process that we see clinically. And that's really been discussed and documented, and how we can actually explore that in many different ways. The second thing, I guess, to point out is that, of course, as a lot of us know, that there's a lot of variability in how people present, and how much of the upper motor neuron systems and the lower motor neuron systems are involved is really quite variable. And we don't fully understand how this translates to how the disease progresses and prognosis, but also this gives us another problem of really having difficulty understanding how much of the brain is involved and the upper motor neurons involvement in each of these different subtypes. So this difficulty and the difficulty of really understanding clinically the brain involvement has led to lots of critically unanswered questions. I've just listed a few of these here. But these are really, I think it's important to iterate that to do more than just treat symptoms, we really need to ultimately find a cure. Of course, that's always our goal. And our best chance to do that is to better understand underlying cause, that we can target our treatments in a more successful way. And we know that this process is a multi-step process, and all the multiple upstream effects that Rosie also talked about that might lead to a common pathway of degeneration. But we really need to try and understand what the earliest problems are that go wrong, because the earlier points are the most likely to be successful when we're trying to develop new treatments. And the most important reason for drug failures up until now is really not understanding these basic disease mechanisms. And with the brain being such a critical part of this picture, this has been one of the major hurdles that we're now trying to get through. So up until quite recently, what happens in the brain could only really be based on clinical observation. And we know from clinical observation that at the onset of the disease, there is generally a very focused onset. So we either get symptoms in an arm or a leg, or bulbar symptoms of vocalization. So the hypothesis is that there is a kind of focused area of the brain where the disease starts. And then from that, there is a spread of disease as we see clinically. And finally, we have kind of a spread across both hemispheres and through the motor system. But this again was a hypothesized model. This was a model that was suggested in 2009, really again, because we had very limited means of actually really understanding what was happening and going on in the brain. But compared to kind of drastic techniques of the 19th century, we do now have some more advanced research techniques and tools to look at the brain in vivo in people. And the two that I'm going to talk about are two that I've been looking at in my clinical research. The first one I'll talk about is some advanced neuroimaging, and particularly MRI techniques that we're using to really look at the brain in far more detail. And the second thing is looking at neurophysiology. And Rosie did discuss this in the mechanisms of excitatoxicity, but really what we're trying to find in there and how the two things tie in to help us understand what's happening in the brain in MND. And this is work that I've done in Sydney through my PhD, but also in Oxford for the last 3 years with the MND team there. So just going back, it was really not until the 1970s that we had one of the first scans that eventually would lead to the MRI brain images that we have. And if you compare that to the extraordinarily detailed images of the brain structure and function that we can see now, you can really see how far we've come in terms of advancing this technology and trying to understand exactly what's happening in this area of the motor system. And the MRI scan, I mean, lots of people would have probably had an MRI scan. And essentially what it is it's a large static magnet, and it utilizes magnetic fields. We measure that in Tesla to produce very detailed images of the brain. So to give you some perspective, for a standard hospital scan, that's usually 1.5 Tesla or 3 Tesla. The strength of the magnet is about 1,000 times the strength of a typical fridge magnet. And we know that is, the stronger the magnet, the better the detail of our pictures that we can use. And these are the kind of standard sequences you'd have if you had to go for a scan in a hospital. And we know that there are several different types of sequences you can have in the MRI. We can do sequences to look at the structure of the brain. We can do sequences to look at how the brain is functioning. We can look at the chemical signature of the brain and see if that's abnormal, and compare that to people who have a healthy brain. And we can also look at how networks in the brain are connected, how different regions of the brain are talking to each other, and how these are interconnected, or whether there's any abnormality in that. So what we really see is a kind of big picture viewpoint. So kind of in comparison to the microstructural detail that we heard in the last talk, this really gives us a different overview of the same problem. As you might be aware, and one of the biggest problems and reasons why this is the area of research that I'm in, is that the standard scanners that we get in the hospital system, so 1.5 Tesla or 3 Tesla, is often found to be normal. So despite there being a clear problem in those motor pathways, we don't often see a lot to justify that on an MRI scan, the standard MRI scan. And that is just a limitation of the tool that we use, and also the complexity of the disease. And so currently, when we use MRI in the clinical process, we're using it to rule out other diagnoses in the cases of MND when we need to. And so you can think of the 3 Tesla scanner, which is the hospital scanner, really like your most efficient, cost-effective method to look at images. It gives us pretty good details. It rules out the important things and nasty things that you might want to rule out. And in general populations, this is kind of an adequate scanner to use. But of course, for MND and the complexity of the problem we're talking about, we really need to advance our technology. And one way to do that is increase the strength of the magnet. So one of the things that I've been looking at in particular with the team in Oxford is using a 7 Tesla magnet, which is what you can see here. And that's kind of like upsizing your car into something a little bit more fancy. It's a little bit more expensive, but it gives us better performance, and it certainly gives us much better pictures. Essentially, as I said before, the stronger the magnet, the more clarity the image has. And to put this magnet in perspective, this is about 5 times or so stronger than those giant magnets you would get in junkyards that can pick up your cars. And this magnet could certainly pick up a double-decker bus. So it's a very strong, powerful tool. And they're actually, just for completion, there are 9.4 Tesla scanners. Now, this would be kind of a supersonic type of vehicle, I guess, but this really comes with a lot of cost and a lot of technical difficulties, plus a huge team of engineering that needs to manage this. So really, the 7 Tesla is a really good compromise for the problem that and for the actual tools that we need to use. So it's still quite an exclusive club, though, the 7 Tesla scanner. So this is just showing you the worldwide numbers of scanners that we have in 3 Tesla and the number of scanners we have in 7 Tesla. And you can see that there's about, I think, about 10,000 scanners, 3T scanners around the world. But you can see in 7 Tesla, there's only 2 scanners really in Australia, one being at the Florey in Melbourne, where I am, and there were 2 scanners that I worked with, 1 scanner that I worked with in Oxford, but 2 scanners in the UK as well. So these are really advanced technologies and advanced techniques, but they do give us kind of exciting possibilities to really try and get into the detailed structure and function that we couldn't explore in a way before. So there are a lot of advantages of using this technique, but the one thing that I wanted to say in particular, and one of the kind of most frustrating things, I guess, when we're doing a lot of clinical research and we want to try and find the answers, is the time that it takes. And one of the good things about using these advanced technologies is that you need much less people to get the same conclusion and to have robust and accurate outcomes. So this was a study that they did in the UK, where they looked at the same question in a 3 Tesla scanner versus a 7 Tesla scanner, and they needed quite a reduction in the number of subjects and less time to actually get to that conclusion. So it's another kind of benefit of why this technology is really important. And one of the other things that I'm really working on is trying to increase resolution of the scans that we can see. So like in a camera and the focus of the camera and the pixels that you can get, we can really try and refine, even in a 7 Tesla scanner, we can refine the detail that we can get. And this just shows you different types of resolution. You can see from the kind of less resolution, blurrier picture to how sharp we can actually get that image. This all comes out with, technical difficulties, but this is something we're really working on, again, to really see that fine-grained detail of what is happening in the brain in MND. So what can we actually see with the MRI, and what have we seen so far with the MRI and these kind of advanced scanners? So firstly, just looking at brain structure, we know that we already kind of know even from the 19th century that there's definitely some changes in the brain when we look at it in the primary motor cortex. And when we look at this particular image here, you can see these A, B, and C are images, MRIs from patients, and D, E, and F are MRIs from healthy participants. And particularly what they're looking at here with the arrow is that you can see in this particular region, which is the hand knob. So this is the area that's responsible for when you move your hand. This is the area that really represents that. There are some differences here that you can see, that you can appreciate compared to in the healthy patients, particularly when you look at kind of the dark line here underlying that hand knob, so hypoattenuation. And those are the kind of changes that we can see on a 7 Tesla to really allow us to locate the abnormalities very focally. But it also tells us qualitative information. Is the brain tissue structurally abnormal, or is it not structurally abnormal? And these are the kind of detailed questions we want to try and ask. The other thing that we can do in MRI is really look at brain function. One of the most important things that we do. So like we were talking about moving our hands and doing simple tasks, this gives us a way to actually literally watch the brain while it works. So we essentially get someone to tap their fingers or do something in the scanner. It doesn't have to be this, but in the bottom left, you can see that there's some increase in brain activity that happens when you do that. And we can actually watch that happen in real time. And for this example, you can see in that particular area, which is responsible for the hand movement, we often see some changes in that area that light up as you actually watch it. So this really tells us about function rather than structure. And this is a really important aspect that we've looked at in terms of understanding what is happening in the brain. And so what actually have we found, and what has been found by other groups as well, is that tying in with what Rosie was talking about, excitotoxicity and overactivation in these areas of the brain, that earlier in the clinical disease, we see hyperactivation. So there's more activity than what we'd see if in healthy controls. And what you see here is in the healthy patients here in the top left, when they're moving their hand, their activation happens on one side of the brain. And you can see in patients, which is in this middle picture here, activation, even though they're moving one side of the brain, the activation's happening on both sides. So there's overactivation of the areas of the brain, even though they're performing the same task. And that comes back to what Rosie was talking about in terms of excitotoxicity and loss of these interneurons, or these kind of balance interneurons that usually inhibit those extra activities. So that kind of ties in again with that function that we're talking about that we see. And later what we see in the brain is that there's hypoactivity, and that really matches with when structural changes keep progressing, there's less activity. So that kind of is a dynamic change that happens earlier on, but that we tend to lose as things progress. And another way to look at that is to try and look at the chemical and the metabolite fingerprints of the brain. And that's something that has been really tried at the 3T level, which has been difficult and shown some conflicting results. And at 7T, this has been tried, and this is something we're really working on, to really look at those chemical imbalances and to see where we're talking about glutamate and GABA and all the other balanced neurotransmitters that give us that balance, to really understand what the differences and the abnormalities and what the imbalance actually is. So this is really an area of work in progress, and something we're technically still trying to develop, but this is really something that's exciting and will be important when we talk about targeting treatments and trying to find out areas where we can actually try and modulate this change. So the kind of other thing that I want to talk about, or the other way that we can interrogate the function and the neurochemistry of the upper motor neuron in a non-invasive way, is using transcranial magnetic stimulation, TMS. And Rosie has touched on this as well. And this is really what I did all of my PhD work on, really to try and understand how these inhibitory and excitatory networks really work in the brain in MND. And we know, as we've already talked about this in the previous talk, but this technique really gives us some magnetic impulses in a particular area of the brain, which we can then record depending on whether we're looking at a limb muscle, whether we're looking at the hands, or whether we're looking at the legs. And we can look at different aspects of TMS, but the major thing that we're looking at, and the major thing that we know, is that in certain conditions in healthy people, and we can see that in red, we see this response in red where the SICI, or the kind of short interval intracortical inhibition, which is the long way to say it, is really this is a normal response in a healthy kind of patient. But what we see in patients in ALS, in the blue line that you can see there, is that there is a lack of this response. So there's a loss of that inhibition. And this loss of SICI is a really consistent observation. And even despite the different types of MNDs or the different clinical patterns people might have, this is a very consistent observation. And it also predates the onset of symptoms, as Rosie said, particularly in genetic carriers of the disease. So this again supports the loss of those GABA transmitting interneurons and unopposed activity of those interneurons and firing of these cortical neurons is again a hypothesis of excitotoxicity. So TMS is really something else that we can use to look at that neurochemistry. So the other things that we've been doing is really trying to bring this into the clinic and be something that we can use for patients in the clinic to really understand how things start in terms of the neurochemistry and the excitotoxicity, and also how things evolve, and particularly to try and understand how different drugs might respond to that. We know that riluzole, for example, when we give riluzole, there is a change in excitability. It actually normalizes for a short period of time. So we know that we can use these techniques in some way, or we can at least try and develop these techniques to be markers of disease activity and also markers of how well drugs may work. And this is something we're still working towards using in the clinic. But the other thing that I wanted to just discuss in terms of what we found in TMS, what we found in this research, is that one of the things is what we can do is, and what we know in the brain is different regions of the brain are attributed to different areas of your body. So for example, this is the area that we know in the brain that is responsible for the hand. And for example, this is the area that is responsible in the leg. So we can interrogate these different pathways depending on whether we want to look at the pathway to the hand or whether we want to look at the pathway to the leg. And what we saw when we did this is that we saw the burden of changes is really tied to where the clinical onset was. That really meant that people who had a limb onset form of MND seemed to have most of their excitotoxicity or their overexcitability in the region, the hand region, and people that had leg onset, for example, MND, seemed to have more excitotoxicity in their leg region. And this really suggested to us that there's a focality of this problem, that really this doesn't spread in a way that doesn't go along with clinical symptoms. This really does go along with your clinical onset region. We also saw that in a third of patients who we were able to study very early on in their clinical disease, that they only had abnormalities, cortical abnormalities in that region of clinical onset. So this was really suggested to us in a number of different ways. We were able to see with this research that there is, according to this hypothesized model that I showed you before, that this really does support this, that there is a focal region where things might start in the brain. And then eventually, as things progress, as clinical symptoms progress, we get a more widespread region of involvement, and we certainly get a more widespread region of excitability that we see with TMS to support this. So this really suggested to us that what we're seeing clinically is also matched with what we see in the brain. And up until very recently, that has only been something that we could hypothesize and not something that we could actually really support in this way. So this is something I think that was really important for us to understand in terms of how things start and how things progress, and something we published a year or two ago. The other thing is, of course, I talked about clinical heterogeneity and this being one of the major barriers to understanding what's happening in the brain. We also were therefore interested to really finding out, is there any changes, is there any differences in our excitability, in our function, in the brain abnormalities that we can see when we look at different onsets of this disease. So bulbar onset versus upper limb onset versus people who have lower limb onset of motor neuron disease. And what we found is that there is a slight difference. And what we see is that probably the cortical hyperexcitability is greatest in bulbar onset disease. It's not by a huge margin, but it does suggest that there is some variability in this, and that perhaps this excitability does drive some of the things that we see in terms of how the disease progresses and the differences that we see between patients. So overall, kind of to answer this question of what happens in the brain, in the human brain, and through our clinical research, is that we really support through MRI, these advanced techniques in MRI, and through kind of focused research with TMS and neurophysiology and looking at the function of the brain, that there are these areas of excitability that then spread to involve other regions. And this is, you know, how can this really help us understand what we should do and how do we kind of progress from here? Well, really understanding these really early disease processes can give us a critical trigger in the pathogenesis of this disease. What we really want to know is, how does this start and how can we stop it? And we can also, you know, use this in clinical trials as markers of the disease to see if things are working and if things are being modulated well. And we can also use our understanding of how this starts in terms of the focus of where it starts to try and better target things and target our therapies in ways that we haven't done before. So this is kind of ongoing work that I'm doing with these advanced systems, trying to study the motor system using these techniques. And we're doing that now in Melbourne, and I have ongoing work with Oxford, but we're doing this now in Melbourne with all patients with MND. So of course, if you want to get in touch with us, please do feel free. And I'd just like to thank all our collaborators in Sydney and overseas, and our patients and families and the funding bodies. And thank you again to MNDRA for asking me to speak.
Awesome. Thank you, Thanuja and Rosie. They're two fantastically complementary talks. They're looking at a similar problem from two very different approaches. We've got some fantastic questions coming through. First one up, I think perhaps for Thanuja. So are there, you know, a lot of patients when they first start having problems, a stroke might be suspected, and often I think people with stroke get an MRI. Are there any sort of telltale signals from MRI that you could go, actually, no, it's not stroke, but perhaps it's MND at the moment? Are you that far along in those studies?
Yeah, I think that's a really good question. I think this is one of the kind of things that we always find in the clinic, to be honest, because we often have, you know, when people do develop a problem with their limbs or their voice, stroke is one of the first things that is thought about. And there are definite ways that we can tell. So, for example, a stroke is usually manifests on an MRI, and actually can manifest on a CT scan as well. So that's something that can be told very early in the piece. But also clinically, it's a very different very different type of presentation. So that is something that often can come as a disease mimic, but there are definite ways to tell the difference between them. And that's another reason why we often have MRI scans as part of the diagnostic process, not for everybody, but just for some people, just to exclude some of the other causes that might mimic cases of MND. The MRI scan and the standard MRI scan is good at ruling those things out. And
I mean, there is a lot of sort of speculation out there about contact sports as well being obviously an influence on developing MND. Is there any evidence around sort of scanning of brains that could help with that, answering that question?
Yeah, I mean, I think that's always a question that comes up, physical exercise and contact sports, and that's certainly something that has now become more of interest with the big projects going on with football and chronic traumatic encephalopathies. That is certainly something that we think about when we think about the multi-step process of this disorder. We know that it's not just our genes that's contributing to this, that there's other lifestyle environmental factors that we know happen, and small things in small ways contribute to a final common pathway. So all of these things are very important and things that we would look to. But kind of when you think about the population, there are so many different factors and variabilities that people go through in their own life that when we look at that in together, it's really difficult to prove that one thing or another might be a cause of MND, and that's always the barrier that we have. So all of these things, one of the things that's happening at the moment is that there's a big registry called MND, where everybody can put in their data and their information, and that can really help us understand what the common things are that people experience that might be contributors in this way.
Yeah, and if I could second that, I encourage everyone to sign up to the MND database to make sure we get as much data in there as possible. It's a question for Rosie, assuming you're still there. Yes. You're muted as well.
Definitely still there, just taking notes on Thanuja's lovely talk.
So we've got a couple of MPY questions. The first one, I guess, is the killer question, is the chicken and egg question. So are the MP levels higher in patients that have longer survival just because it increases with damaged neurons, and it's just like an effect rather than the cause, or are the people living longer because the MPY levels are higher?
That's a brilliant question, which we haven't been able to tease out yet, and quite possibly it is. And if it is, that actually points to quite an important mechanism still. So if excitability changes and this MPY peptide is changing with it, to try and, I guess, keep those excitability levels in check, and it's not able to do so, why not? And that's actually ongoing research that we're doing at the moment. So we've been doing a preclinical trial looking at this MPY compound in its native state. If we apply it to disease models, what effect can we actually see on the disease progression and pathology? And what we've been able to see so far is that when you apply MPY, you can actually modulate the disease progression in animal models for disease. So it's again pointing towards, I guess, more evidence, much like Thanuja's work, that mechanisms that modulate that excitability are going to be very essential to understanding how we can slow and halt disease progression in MND. And in terms of if MPY is tracking with excitability and is going up for people as long as they live, you know, there's a number of different pathways that we can consider as important, because we have agonists and antagonists that we can use, different drugs to modulate this pathway. So that's not exactly a line in the road as such. It's actually quite an interesting finding, and it can lead us towards more ways that we can actually understand to modulate that excitability.
So have you tried those studies yet? I guess there's a bunch of mouse models out there that have altered MPY levels, or, you know, the various parts of the MPY pathway. Have you managed to cross those onto a mouse MND model to see if it affects the outcome or not?
We've done one better, actually. So we thought about going down that route, but we thought about what's something that's going to be therapeutically, I guess, mostly relevant for people with MND. And there's been some interesting work coming out of New York where people have been trying to apply that peptide in people with post-traumatic stress disorder. And they've actually been doing this via nasal application. So just like you would with some sort of allergy medication, where you use a nasal administration through just aerosolation of a compound up the nose, and then that drug then gets in. So what we've been trying to do in an ALS, I guess, animal model is use a nasal delivery route to apply that MPY and see the effect that it actually has on survival in that animal model. I haven't published this work yet, but writing the paper at the moment, and basically what we can see, very, very excitingly, is that actually nasal delivery can actually change, I guess, the survival in that animal model. So it's going to come down to how specifically, what modulators of that MPY pathway we utilize. But that's something that we're always considering when we're thinking about how we can, I guess, learn what this pathology is doing, but also how can we actually target it, and in a way that's actually, you know, achievable and therapeutically relevant for people with MND.
So that drug is already out there, is it, as an approved treatment for other diseases? You said
not currently. No, it's reached, I think, phase 2 clinical trial for post-traumatic stress disorder. But I don't think it's being utilised yet. They, I think, as far as we're aware, show no off-target effects and no adverse events, which was that initial work. And I think that group is still working to see how it can possibly help for people with post-traumatic stress disorder. So a lot of the groundwork that is very fundamental to translating, I guess, laboratory-based research towards in the clinic has actually been done for this compound, which is another reason why we're quite interested in what it's actually doing in the disease.
Okay, cool. So I guess one big question out there is, or question, whatever, is riluzole. Obviously, that targets excitotoxicity. A question for both of you, this one. I think, to me is, if riluzole can, inhibit glutamate, inhibit excitotoxicity, how come it doesn't have a greater effect? Can either of you sort of...
I can speculate. Thanuja, if you want to go first, I've got some thoughts on that myself. But I'm happy to go. So there's a number of reasons possibly why. As someone who's very interested in the inhibitory system, for example, if that excitability and inhibitory balance is required to maintain, basically if you get progressive loss of that inhibition, you're just exacerbating it and getting worse and worse. So riluzole is only going to work for so long, and possibly only to the receptors that are viable. And then, so if they're intact, they can function. But if the disease progresses beyond a point, perhaps it's not having its effect. And inflammation could also have a role in that. So we know that in MND, there's a progressive change in inflammation that also occurs in the MND brain. And I'm quite interested in how that inflammation also might be affecting that inhibition and making it worse, such that there's more and more of this glutamate overload occurring, and maybe it's not having its effect. I mean, the alternate hypothesis is this changes in activity in the brain that are lost over time, and so the riluzole can't work anymore because the brain can no longer respond. I don't know if Thanuja had thoughts on that.
Yeah, no, I think, I'd echo a lot of those thoughts. I think what we've seen, of course, in neurophysiology is that, and using TMS, is that riluzole modulates the excitability for a few months, but then actually you go back to being excitable, or hyperexcitable. So we know that there's a kind of transient effect, and really the question about why that is transient is still, I think, unanswered. The other thing I think we think about riluzole is it's primarily originally an anti-epileptic drug. And we need to kind of understand a little bit more about how to target these drugs in a kind of fashion, as we kind of saw in both of the talks of both what we talked about, really, is that everybody's slightly different. And what we also see with the riluzole experience is people have different experiences with it. And we have, not a lot of evidence to say exactly what stage of the disease riluzole works in. So there is lots of papers that suggest riluzole works very early in the disease, and there's some papers that have come out more recently suggesting that maybe riluzole works later in the disease. So that's still something that's questioned. And I think, again, like you said, Rosie, when things are working later in the disease, we have less of population of neurons to really work with, and therefore it's a much harder thing to have benefit from when things are working late. So I think there's a lot of things we still have to understand about that. And also, it also comes down to, I think, understanding the differences between people and the different subtypes, because unless we really understand what is driving those differences, it's very difficult to understand how these drugs are working, and also to make sure that we can benefit the people that it will benefit, and that we're not missing out in subgroups that actually might have a huge benefit in one drug and not the other. So I think riluzole is a good example of that.
Great. I think we've got time for just one more question. This is a question that's come up just about I think the ketogenic diet has been shown perhaps to affect glutamate levels. Have you any idea if that has any role or effect in MND? If any of you any idea about that?
No, sorry, I haven't done much reading on that.
Yeah, I think there's been certainly this comes up a lot in clinic because, it makes complete sense because we talk about glutamate, we talk about, hyperexcitability being driven by glutamate. And of course, then, the next logical thing is, well, can we actually modify that in our diet? And unfortunately, the evidence so far suggests that that's kind of too much of a simplistic view, I guess. If it was something that easy, then we'd all celebrate, I think. But what is actually happening in the complexity of the disease doesn't really translate to just being able to modify glutamate in our diet, unfortunately. So we have to kind of try and do that in a different way. And really from a diet perspective, I guess that also raises the point that some of these diets, certainly what we see is that people do go on different types of diets to try and increase their nutrition and increase, anything that might help with excitotoxicity. And the major thing is really that we know that weight loss is an important thing in MND, not to have weight loss, that is. And your metabolic function is very important. And that's one of the things that is really important to maintain. So we always kind of suggest, a kind of high calorie protein diet and not to try and do anything that you kind of really lose any dramatic weight, because that would really have an adverse outcome.
Great. I think we've probably all got a million more questions, but unfortunately we've run out of time a bit tonight. So I'd just like to say thank you again, both of you. That was fantastic, and it's really brilliant insights. I hope everyone else has learnt as much as I have tonight. So thank you to the audience as well for watching. Everyone will receive a link to the recording, so feel free to distribute that across all your networks. As many people as possible. We want to hear about this brilliant research. And also just a bit of a plug for one of our programs. We're launching this year the Clinical Research Learning Institute, which is a program where we hope to sort of teach members of the MND community about research, and hopefully they become advocates to help us talk to government, talk to whoever needs to be told about the value of research into MND. So please go to our website, to the MND Australia website, and look up the CRLI, the Clinical Research Learning Institute, and see if it's something that you fancy doing. It's basically a 2 half-day course where we teach you the real basics of understanding research, statistics, how to read a research paper, how to advocate for support, etc. So we'd love to get as many people as possible signed up for that. So that's it. Thank you, everyone. Thank you again, Thanuja and Rosie. And good night to everyone. Thank you.
Thank you.
Thanks.